Hello.
Hello, how are you?
I'm pretty good.
Good.
Managed to plant a garden and stuff.
Oh, good. Yeah, that's great.
It's
Yeah.
called planting it right away after you rototill.
Oh, right.
One year I chased the rototiller planting the garden. This year wasn't quite as bad, but almost.
Yeah.
It's on the edge of not being able to do these things.
Yeah. Well, good luck with that.
Oh, and I got four new baby axolotls that are 6 months old, and so by Christmas they should be a year old.
Oh, good. Good.
Yes.
Yeah.
And I put Winnie with Chomper and Snickers, but nothing's happening.
Oh.
It's June. I don't know. See, I am trying for eggs, but it's
Yeah.
not working.
Yeah. Hello, Dick. Hello.
No, you're off. Microphone's off.
Oh. Yeah.
Well, maybe a virgin birth at Christmas.
Yeah.
Hello, Wataru and Harikrishna. And Tom. Hello. Haven't seen Tom in a while.
Hello, Tom. Been seeing you for a while.
And Jesse's here. Hello, Jesse. So, welcome to the meeting. Karan said he might be able to join a little bit later, so that'll be good. So, I guess this is the third week of third meeting since we started our GSoC, had our announcements for GSoC, and so this is the community period. Harikrishna and Wataru, did you have any updates for us, the questions that you might have, things you wanted to discuss?
Hi.
Hi.
I discussed with Jihan how to start the project, and we decided to start with the deciding what inputs and output formats we expect in each modules. So we are going to start with check what outputs the Devo learns and other libraries like DevoWorm provides.
All right. Well, I'm glad you two were able to get together. I know I told Jia Hong last week that we needed to get together on that, just so you don't replicate efforts, and that you're, especially with this project, because it's something, it's like, I know we've been talking about the spherical map for a while, but this is something new, and I'm not quite sure how it's going to turn out, or, I'm not quite, I mean, I know what the goal is, but this is something that's going to be very trial and error. So thank you for meeting with him. And did you have any questions about any of the materials that I posted in the Slack for the community period?
No, for now, I don't have any specific problems. So yeah, I'm still catching up.
All right. Yeah, and this is a good time, like the community period is a good time to get all this straightened out, like all the things that you need to do to get started on the project. So anyways, yeah, if you have any other questions, put them in the Slack, and I'll look forward to talking to you about it. So Jesse actually is going to be a co-mentor for the GNN's project. So it'll be Wataru and Hong who are going to be in that project, and Jesse's works with me on some other things GSoC related, as well as a bunch of other projects that we're working on. And so he's a lab manager, so he's good. He's good at sort of organizational stuff. He's also very good at done a lot of stuff with like computational neuroscience and things like that. So he can try to help you with some things. I wouldn't, really technical stuff, maybe not. I don't know how you feel about that, Jesse, but definitely, if there's a question, if there's some, something you need to find out about, you can get in touch with me or Jesse. And then, we'll be planning other things, especially during the coding period, to help you work on these projects. So, just to let everyone know about that. Okay. Harikrishna, do you have an update for us? Okay, Jesse said yes. As Bradley said, I won't be able to talk much today because of background noise. Yeah, so that's fine.
Hello.
Hello.
Yeah, so so last week I tried to go forward with the enough algorithm. I also talked with Karan. So right now we are working on that. Like, how do we use it exactly for the embryo problem? So that's the update of it.
All right. So how are you coming with data? Are you all set with data, or are you needing different data sets, or I don't know how that got resolved.
Right now I have some of the Pepper data sets and the Pearl data set which Susan had sent. I'm using that.
Okay. What's that? Susan has holding up something. I don't know what it is.
Yeah, these are, I got these from All Holes Perforating, and they're spherical holes. So I'm hoping to put them on my ball bearings or something, and just put paint on there, and then move to another spot and put paint, so that there's some proper pattern. So I've got two sizes. So I'll play with this. I've sent for some ink, that resin alcohol ink, which is apparently better for not running. So I can do that, and I could also use larger spheres for some of this, so that may be easier to paint something on. With a larger sphere is all right, isn't it? Because you're just looking at a sphere and trying to match it. So I even have some paintball balls that are 6 millimeters that might be easier to use.
Okay. Susan, you could use that material for sievings.
Yeah.
One
is too small, but the other one is.
Yeah, the local gravel might have enough texture, so you don't have to bother with painting.
Yeah, well, I'll try all kinds of things here. Willing to try things.
Yeah, and this is for what is this for?
Well, taking pictures with my ball microscope to try to get something that you can actually match, so that you can image it.
Okay.
Because these things are just very uniformly white.
Right.
And
yeah.
so they're
Okay.
not. It's not.
No reference points. Yeah.
Yeah.
Yeah. So if you trying to get texture on a 2 millimeter diameter sphere is rather difficult.
Yeah. Put them under the microscope, and if you touch a pen to them, the pen pulls the sphere away, and you get all these little blotches, which I gather are kind of useless. So, try something else.
Yeah. Yeah. Wataru, I was wondering if you and Jia Hong had discussed your data sets that you're going to use. Do you have enough data, or how are you faring with that?
This sets. Yeah. We are now checking the datasets you gave us,
Okay.
and yeah, we don't have any specific datasets to. We haven't decided which datasets to approach yet.
Yeah. That's good. I just wanted to make sure that you had something that you thought you could work with. And in past years, people have used they've used, well, the ones that I sent you links to, those are usually sufficient to do benchmarking and things like that. So, there's the embryo that you have, and it, the cells divide over time. So you have these movies, and then each movie has a frame, and the frame has like a certain number of cells in it. Some of them are dividing, some of them are just cells. And then we have information in C. elegans. We have information on the identity of the cells. So those cells can be labeled, with this information. So this is something that is nice, because a lot of other species or a lot of other developmental systems don't have this information. So you have labeling pretty much built into this. So that's good. And then this is again, like, we've done different ways of doing this throughout the years, where we've worked on how to get the thing to recognize cells, and how to set up I think it was 2017. I think we did a GSoC project where we did some work on this, where we were connecting the lineage tree with the images. So it's something that can be done. So there are different ways you can approach this, is what I'm saying. So just let me know if you have any questions about anything, and so that you can you can move forward on it. Okay. Well, thank you for those updates. So this past week, like Friday, I gave a talk at the Google Summer of Code Contributors Summit. So this was for all contributors, mentors, and people running the program. So there were a lot of and I have a link to it in the Slack. I think I put it in there Friday, and it's a live. You should be able to still access the live stream. It's not public, but you can get to it. And so they had a lot of interesting talks, a lot of different topics on open source development, and on being a mentee and being a mentor. I gave a talk on some of the educational initiatives we've been doing over the past several years. So that's an interesting session. I actually met up with, I shared a session with Arnab Banerjee, who worked with the DevoWorm group back in, I think, 2017 or 2018. And so it's kind of fun to watch him as he's moved on in his career. He's now working at Credit Suisse, which is a bank. And he started out in open source. They wanted his open source expertise originally, and then he's moved on to project management and those kind of roles. So that's nice to see him move on in his career. Of course, it wasn't all due to GSoC, but I think that helped him maybe getting his foot in the door. Maybe it's helped him in his career. He was definitely at the Contributor Summit, so they and a lot of people found that a nice, interesting talk. And there are other talks on licensing, talking about how open source licenses are, depending on what you want to do with your software. Some licenses can be good and some can be bad. So, for example, they talked about the GPL license, which is an open source software license. And in corporations, oftentimes these can be somewhat undesirable because they give away maybe too much intellectual property. So, people create an open source project in a corporation and they put a GPL license on it. It can pick up dependencies that are, maybe proprietary that they don't want to share. And so that was something I had not thought about before, but that's because I've not worked in a large corporation doing open source. We had other talks on some of the other aspects of the project or the program. So the program started in 2005 and it was very informal. And they've grown to a very large number of mentees and mentors over a number of organizations that sponsor different groups. So we're sponsored by INCF, and INCF was well represented there. But there are all sorts of other organizations that draw from that program. From traditional open source projects to, I think Wikipedia has some interns. I think like some of the other bigger open source projects have some interns to, actual corporations. So there's a very diverse group of people now involved in this. And so I wanted to share also, okay, I'll share my screen. I wanted to share some things about our, we have a onboarding guide here, actually. I wanted to show, here it is. So I didn't show this last time. Last year when we did Google Summer Code, we made an onboarding guide, and this is the efforts of a couple of people, a couple of former contributors plus myself. Minok Deb, I think, was involved with this. And I was involved in this, and trying to think of who else was involved in this. A couple other people. So this is, it kind of goes through some of the basics of our organization. So what is OpenWorm, DevoWorm, and essential links? So I've talked about OpenWorm before. They're trying to build the first digital life form in an open source fashion. So we have some links here to the organization, the Slack. And this is a couple of talks. These are talks that I didn't discuss before. But this kind of goes through some of the OpenWorm materials, some of the things about using GitHub. Although I don't think a lot of people who usually though we get applicants who are at different levels of knowledge of GitHub, believe it or not. So this is just to get people up to speed on GitHub. But this is more this is an onboarding guide more broadly for people joining DevoWorm. So if they're joining and they maybe aren't really that heavily computational science oriented, they can learn about GitHub. And then, we have our resources for DevoWorm, which is a separate thing from OpenWorm somewhat. We have our own GitHub repository, group website, organization repository, and so forth. And then these are slides that this is Krishna Pillai, who is now working at an open source organization with an open source organization called Tohi, and they're doing a lot of templates for machine learning. So he made up these slides, and they're kind of fun. They kind of walk through open source, why you should contribute, and then giving a little bit of information about our past achievements, and then then after that we have some general FAQs here, and then some links to some of our model organisms. So these are some links to different things. If you're coming into the organization, you want to see what kind of organisms we work with actively. So we're working with C. elegans here. There's some examples of C. elegans. This is the diatom, which is a marine organism. It's an algae, and they live in these colonies. And diatoms are they're actually made of glass. So these are silicate organisms, and they live in these colonies. Not all of them live in colonies, but the ones we study live in colonies, the ones we've been publishing on. So we have a little bit about diatoms here. And then we have axolotls. So this is the project that Harikrishna and Karan are working on. And they're working on axolotl embryos, and there's a axolotl model organism community, just as there's a C. elegans model organism community, and we have some links there. And then we have some biological data sets. So there's some overlap here with the DevoZoo stuff, but I wanted to bring this DevoZoo stuff out and kind of give people some pointers to some of those things. So if you want to read this onboarding guide, I'll post it in the Slack. You can take a look at it. Okay. Any questions so far? I know Tom hasn't been here in a while. Would you like to give an update on what you've been up to?
Well, okay. Yeah, just thought I'd check in and sort of just finished a project, sort of got a little break here. Worked on a little project with, I've never seen this done before, so I thought I'd try it. How like sequential neural networks, how well they perform if you like disrupt the streams, the temporal streams. So I investigated like LSTM and TCN and of course Mechanistic, which is my baby, which did the best, strangely enough. So what it means is, like, suppose you have a task, and it's a stream of like inputs and outputs, like stimuli and responses. And so you learn that, and then what you're doing is, I mean, this happens to organisms all the time. You're going about your business, and something happens, and it's you have a disruption, right? And you're making a cake, and you run out of sugar or something. So something gets injected in that path, or just deleted from the path and substituted from the path. It's a little piece of stuff you know about. It's like a modular chunk, but you have to go like detour to do that, and then come back to the original path. So I thought this would be an utter disaster for most neural networks, but it actually isn't. So I was kind of surprised about that. So just kind of I'm posting that on ARX IV like tomorrow, I think.
Okay.
So I have a little break here, so I'm checking in.
Oh, good. Yeah, this is nice because
good to see the group still going, and everybody and people are here and stuff like that.
Yeah. So you done a lot of work with neural networks and AI. Could you tell people a little bit about that?
Well, yeah, I mean, mostly, doing machine learning related to animal behavior. Even a C. elegans can be modeled as a kind of a cellular automaton, So I guess my little thing is, animals are, they still have lots of capabilities that AIs don't have whatsoever. So they're very flexible, and they deal with changes in some ways that are just amazing, So, I mean, I was just like reading about, bees and stuff like that, how they can count, and, how they remember things, and they have this like group intelligence. That's like all real fascinating to me, so I sort of feel compelled to try and get some of that stuff, find out how some of that stuff might be done in artificial intelligence and artificial neural networks. So,
Yeah, so you created this morphozoic platform, and we've talked about that. If the GSoC people are looking through some of the stuff I sent into the Slack on the DevoWorm open house several years ago, we did a tutorial on morphozoic. And this is
well, I didn't create it, we did.
Okay, yeah. And this is a cellular automata that is these nested neighborhoods. So we did this session on the tutorial, and the that software is still available for people if they want to use it. This is a classic open source software, so you can use it for, if you have an idea. I don't know, maybe it's not very close to what people are doing in these projects, but it's definitely worth, revisiting, because we've been talking about neural networks last year, and then we kind of dropped it because we didn't have, we didn't have a GSoC project this year on it, but it's something. It's still there. It's outstanding. So I've been trying to encourage people. I never really got to it directly, though. But,
well, I'm not saying I'm the world's expert at it.
Yeah,
but there's so many things happening, so. I mean, like I just ran across, like, within the last year, this thing called the Temporal Convolutional Network. It's fairly new. It came out in 2018, so it's kind of it's convolutional, but it does like temporal streams of things that are moving along in time. So it's supposed to be really good. It's supposed to be superior in some respects to the standard, like LSTM has been and then there's a new one which is a bit, forget the name of it, but anyway.
Yeah, we just had a conversation, I think 2 weeks ago, about like having, building networks where they sort of repair, like looking at if you knock a part out and it repairs itself, what's the, how do we measure the capacity for that, or how do we look at the performance of, and I'm not talking about neural networks
even,
necessarily.
That's like, there's this, dropout thing, which is actually makes it better, it doesn't, reduces the overfitting, which is kind of like knocking stuff out. So, but the animals, like I got involved with the Open Worm project, like 10 years ago or something like that. Because animals are, I think it's just like the big piece we gotta try to figure out how to keep working on that, get that into, if we want to actually make AIs real. I don't know, maybe some people say we don't want to do that.
Yeah.
But, like Elon Musk,
Yeah.
It's like, oh no. Who knows? Maybe he's right.
Yeah. Well, there have been some fierce debates.
Fun. It's sort of fun in an aggravating way, It's like the muse speaks to you and then you have to go do it. So, please, not another one, no. Yep, you gotta do it.
Yeah. So, and then you worked on the robots project in OpenWorm, so.
Oh, yeah, the robot. It's still over in Austria. I'm hoping they can keep it over there and it never comes back. I don't know. They pay me like €50 a year now to keep it over there.
Oh, really? Oh, so the last I heard about
that other guy I worked with, the other robot guy in Australia, he's going great guns. He's done some amazing stuff.
Really?
Yeah. Shane.
Yeah, Sean.
You remember him?
Oh, yeah.
Yeah.
So he's kind of doing his own stuff, like soft robotics.
Oh, yeah. He's robots. Pretty amazing stuff, too.
Yeah. I was always impressed
Out
with...
of the Bots is the name of his company.
What is it?
It's called Out of the Bots.
Out of the Bots. Like out of the box,
I don't
but
know
bots.
how he came up with that, but...
No.
Yeah.
Yeah.
So Bradly, you're like, are you still with University of Illinois, or I saw something else you're hooked up with?
Yeah, I've been doing work. I've been doing some open source community management for Rockwire, which is a initiative by the university building campus apps and things like that. So that I've been doing like a lot of the stuff that I like kind of do here, and then working with mobile development and things like that. So it's a little bit different than like OpenWorm, but it's a interesting thing. And I've also been working with my own lab group where we've been doing a lot of stuff with AI and cognition, cognitive science. So that's been a little bit different than this again, but it's, So, yeah.
Yep.
It's always good stuff.
And I see Susan's so up to her. She's got a ball telescope, or ball microscope now, huh? Whatever that is. I don't know what that is.
Susan? She's going to go get it. She's got it. I'm a prototype right behind her there. Yeah.
Okay. Oh, here it comes.
Yeah, there we go.
Oh my gosh. Oh wow. It looks like one of those fusion things. It's gonna
Yeah.
great antimatter.
Like a neuroimaging device, yeah.
Wow. That's okay. Oh, I see all the angles, all those different
cameras coming in.
Yeah.
Is that okay? They call it a ball telescope because it looks like a ball, huh? It's like
Canadian Sputnik.
okay.
Yeah.
Okay. It's not a polyhedron telescope.
Susan, you're muted. I know you've been saying stuff, but
Yeah, sorry, I'm saying, trying to find a small ball that will go in the middle of it, and it's a 3D printed ball, but
that's what you're
in the
talking
middle
about.
of it is supposed to be a salamander egg. So I'm trying to figure one out somehow, because
Okay.
salamanders are not laying eggs, even if I
Oh, really?
males and females
Oh.
together, they're just going, it's June. It's like, yeah.
yeah. Yeah, thanks for the tour of the ball microscope there. It was nice.
Yeah.
Yeah, in my
Yeah.
nasty workroom here. Anyways,
I see Dick still got your treadmill there going. Oh,
Yeah, I still got my treadmill. Okay. Yeah, we ought to talk soon, Tom, because there's a whole bunch of things I'm up to right now.
there we go.
Yeah, I actually looked at the Ising stuff again. It looks like a physicist who invented the cellular automaton or an Ising model first.
So Isaac.
Yeah, I'm guessing that. That's true. Yeah, probably goes way back.
Yeah, it's a funny story because Isaac made a model for a phase transition, but he made it one-dimensional and it didn't work. And then somebody took his model,
Okay.
made it two-dimensional, it gave a perfect phase transition. And Isaac became famous.
Wow. Okay.
In one dimension you don't get a proper phase transition.
Huh. Okay.
Okay.
Yeah,
Two and three
the
dimensions,
paper I'm reading, the one you sent me, I'm rereading it. It's the two-dimensional one. So.
Yeah, the major problem is that there are about 800 papers on one-dimensional Ising models.
Oh, really? Okay.
Yeah, and the one-dimensional ones, if you take them versus time, generate patterns that look like Wolfram patterns. Okay?
Okay.
And this led to the question as to whether or not Wolfram patterns are open or closed.
Right. Yeah.
Okay? So that's where it sits right now.
Okay.
Yeah,
There's no intersection between the one-dimensional Ising model literature and Wolfram patterns, except one paper I wrote on diatoms.
Oh.
Okay?
Okay.
So the physicists are totally unaware of Wolfram patterns as far as I can tell.
huh.
Yeah. Huh.
Okay?
Yeah. That's. Yeah. I mean, when I reading the Ising, it immediately goes. You immediately think of cellular automata. So, but you don't see that referenced anywhere. It's like they're in a silo.
Yeah. Right.
So.
So anyway, it's fun. It's also related to the question of the patterns on snails.
Mm-hmm. Mm-hmm.
Are they really Ising models? Are they really Wolfram patterns with Wolfram rules, or are they something else?
Yeah.
Okay.
Yeah.
Mm-hmm.
So it gets started.
Yeah. I don't think we've gotten anywhere any further on that. So.
No, that's about it.
Yeah, sort of thinking about it. Yeah. But.
Yeah. That's
need
a
a
very
reader.
interesting question.
So yeah. So let's see. Looks like Wataru had to leave. I'm gonna go through some things that I've been pulling up, maybe some things that we've made some progress on. So like I think I've mentioned this. This is a ways back, but I talked about the mathematics of I can't show. There we go. The mathematics of DevoWorm, which was a sort of a collection of diagrams I was bringing up. And I don't know if my screen is having trouble sharing, but let's see. I can do this. All right. There we go. So this is the mathematics of Devo. I've made a poster of it, actually. And this is kind of just waiting for a venue to put the poster in, but I can also put it up on the web, and we can have this as a standard poster. So this kind of goes through the mathematics of DevoWorm. And so, there are a couple of models that have been put out in papers. These lineage tree dynamics, which have come out in a couple papers, the developmental function and spatial complexity and self-organization. These are some other things that are sort of diagrams, kind of thinking about like the basic parts of this sort of computational developmental biology approach. We've kind of done things related to categories and category theory, which is diagrammed here. Some things from one of the papers on developmental game theory, and then some of these differentiation trees and computational development. So this is a sort of a differentiation tree, lineage tree combination here. So this is just to give people an idea of some of the things that are going on. I guess I haven't put the citations in. I'll put them in like about here. But this is, what I kind of wanted to do with this is to make a poster that kind of goes over these different little methods and, points people to papers and giving them a visual of what's going on. So this is something that if you're interested in like contributing to, we can talk about how to, maybe, do some other things with it. But this is definitely something that, last time I showed it to the group, it was just kind of in these diagrams that were scattered all over the place. So if you look at these two figures here, there are these graphs. And these are actually from two papers that we've done in the past here. One was from a paper in 2021 on comparative embryos, and this one was from like 2017 on C. elegans lineage trees. And I'm having trouble zooming in because this is a poster and it's really big, but you can see these two figures. In any case, I have in the meeting, here it is. Okay, so this is kind of going back to the regenerating network stuff. So in thinking about this problem of regenerating networks, and I told you before that was the project that we were talking about, or the idea that we could create these networks, not necessarily neural networks, but like complex networks that regenerate their parts. If you take them off, they regenerate, and kind of measuring maybe what it is about networks that make them robust or maybe give them this property. So that's kind of a hard, I'm kind of approaching this from some of the stuff we've done before, and I'm not really close to being like having an answer for you, but I wanted to go through some of these older papers. So this paper was information isometry, which is this technique that we came up with in the group around 2017. This is where you have these lineage trees, that each node is a binary value, and you can go down the tree and you can go from like left to right, and you can give the cells different identities in binary code. And then you can do things like flip the nodes around, and you can flip the order, and you can compare different trees in terms of their order. So you can do things like measure different cells if they're in different states in different parts of the tree. If they get flipped somehow, you can measure the distance between the two trees. You can do other things. Now, this lends itself to knocking some of these nodes out. So say I knocked out this part of the tree. I then would have a tree that had no values for 0, 1, 0, 1, 0, or 1, 0, 0, or 0, 1, 1. And then all of its descendants would be removed as well. So you could actually compare that with a tree where it wasn't removed, and you could look at the distance between those two trees. Then you can identify different defects in the trees. You can identify like things where trees have been clipped off, different parts of the trees. But then also, if it's regenerating something, it's going to regenerate according to some schema. So you might start down here where it regenerates from 0, 0, 1, and there'd be some additional branching down here that would have its own code, and that would have a distance from, say, like a regular tree that looks like this, like a null tree. And so you could actually measure that regeneration as well. So there are ways you can do that. There are ways you can encode this removal of nodes and addition of nodes, and have a computational representation of it. That still doesn't give you a mechanism, but one of the nice was that yeah.
I think there's some literature that might weigh on this, right with the nematodes. I recall that there's a bunch of literature on the trees having to do with the vulva of nematodes.
All right.
Are you familiar with that?
I don't think so.
Okay, just search C. elegans and vulva, VULVA.
Yeah.
Okay.
Yeah.
They often give trees, as I recall, and they use different mutants, so they have alternative trees.
Okay. All right, I'll take a look at that. Yeah. And so this is for the entire C. elegans. So the vulva is down one of the lineages. This is just actually based on C. elegans, this example, and it just shows like this binary division. So in some organisms you don't have this kind of binary division where you have an identity. There's proliferation of cells. There's they don't have strict identities. So this is just kind of like a C. elegans like tree, but there are areas like the germline and the vulva and things like that where the tree behaves a little bit differently. But one of the cool things you can do with this then is to look at if you compare two trees and you find the distance between them, you can actually generate random trees, and you can look at this like a biological tree. So you can take a tree from like an embryo, and you can also generate random trees, which means you just generate nodes anywhere, and you compare them across individuals or across trees, and you can actually get graphs like this. So what this does is it shows what they call the Hamming distance, which is a measure of the distance between two strings. So if I have a string that's like, with one binary value and a string with another binary value, and there are two positions that are different in that string, then that's a Hamming distance of 2. So if you're familiar with NLP, sometimes this is there's a Levenstein distance. It's very similar to this, but the Hamming distance came first, and it's like they've used it in information theory for many years. But this is a way to evaluate binary strings. And so what you see here is you see that C. elegans has a greater Hamming distance across lineage tree level. So lineage tree level is how many branching events do you witness from like a single cell. So a lineage tree of level 1 has a certain distance, level 2, level 3, and so on. And so the deeper the tree gets, you get a larger Hamming distance between the samples. And you notice, though, that with the random trees, you get this, it's much lower than the biological case. Why that is well, it's probably because in the biological example, your lineage tree has some structure where, you know, there are different areas that are invariant and maybe different areas that get turned, get flipped. So this is explainable by virtue of not having a fully random sequence, that certain parts of the sequence move around more than others. And that makes sense when we think of lineage trees as being these things that sometimes it's important for things to be maintained in them. So what does that mean about like cell divisions? Cell divisions then, you know, we're clipping parts of the tree out. So that means that your cell divisions, you know, if you take out part of the tree, you know, you can represent it using that kind of graph, and it'll give you a representation of maybe, you know, where you can identify these different places where maybe they're more vulnerable to being cut off, where they're more likely to be regenerated or something like that. I mean, I haven't thought it through any more than that, but that's what I've been revisiting this paper and getting that out of that stuff. This is the other paper. This is the one that was published in Biosystems, like I think last year, and this one has a graph in it where it basically it's not the same graph. It's a similar graph, if I can find it. Well, I have it here. So this is, oh, that's not it. This is it. So this is a similar graph where you have the number of divisions versus developmental time. So this developmental time in this graph is very similar to the lineage tree level in this graph. It's just a different way of expressing the same thing, but it's over time. And then you have this number of divisions. And if you notice this top blue graph here, this blue line it looks like it's just kind of stepping up, you know, deterministically. And that's because this is something that is representative of a lineage tree that's dividing in a normal interval. So it's dividing, making those branches at a normal time interval, and it's making a number of divisions over time. So you can see that it's a straight line. There's nothing unusual. I mean, it's actually unusual in biology to see this, but this is like a null tree basically. But you notice that if you use other distributions, you use these other distributions like a exponential distribution or Poisson distribution, and you can generate these computationally. These aren't biological, but you get these different step lengths. So you get a larger number of divisions per unit developmental time. Sometimes you don't get any divisions over a long period of developmental time. What will happen if you use different mathematical distributions is that you can stretch out these steps between the number of divisions and developmental time, and there are different ways that this manifests itself. They all basically end up at a similar point, but there are these different these different step lengths. So one of the things I'm thinking about here is this is something that can be exploited in a system that's regenerating. Do we need to understand some of these alternate distributions as a means for providing maybe a signal for vulnerability or a signal for robustness that would encourage regeneration, or what does this give us? Now, this isn't biological, but what's interesting is that these alternate distributions may connect to some different types of biology. So if we build trees that have variable, the branching is variable in time, where it's different in different parts of the tree, it's variable. That signature then may tell us something about its ability to regenerate, or we can run an algorithm where we actually take out parts of the tree and regenerate parts of the tree and see how those behave, and then understand them in terms of this sort of framework.
Bradley?
Yeah.
I've got a question on that.
Yeah.
Did you consider the model which is common in observational biology, that the time interval between divisions gets longer and longer as an embryo matures?
What was that again?
if you consider how, I don't know what you mean by developmental time there. But if you consider a normal embryo in real time, the time between divisions gets longer and longer for most embryos.
Yeah. Okay.
Take into account here.
Yeah, it's probably something. Yeah. That. Well, that's something to take into account. But the thing that I found interesting about that was sort of the intervals, as they're not uniform, that they have this non-uniform property, and that might have some bear some role in like.
What is the relationship between developmental time and real time?
Developmental time is basically real time. It's just breaking it up into parts. So if you have like 500 minutes of real time, it's like so many units of developmental time. It's just another way of saying real time, though.
Okay, but you're assuming that divisions per time per cell are about constant for all of those?
Well, in those simulations, I set them to be I set the uniform case to be constant, but then the other simulated cases, I showed that they were, not constant. But now you could put that turn that into a biological tree and give it that property where divisions take longer and longer over time, or whatever you want to do.
Okay, I'm just saying it might be interesting to generate such a thing and add it to those graphs.
Yeah.
Hey, Bradley, I know you've been working on this for quite a while. I was just wondering, does when the thing subdivides and differentiates, is there like an entropy aspect to this where like top level divisions tend to separate into chunks that are less, it's like, because I kind of bridge things, there's like a decision tree where it reduces the entropy maximally at the top and goes down. So the subcells of the top level division, do they give rise to things that are like, the complexity of those substructures are maximally less entropic, that sort of thing? Does it? I wonder if nature does that,
You mean like that?
Tends to divide things into chunks that are simpler,
Right. So they'd be less subject to entropy, or
yeah.
If you take, I didn't show like local measurements. So the Hamming distance is sort of a measure of global entropy for different levels, but that's across the entire tree. So if we were to measure like subparts of the tree, you'd probably get this sort of you'd probably get this sort of signature of less entropy over time, just simply because it's there's that dependency, right? At least with a C. elegans like tree, where you get like a subtree, and then that subtree can't really be configured too many different ways. But this is sort of independent of function too. So if you were to like take a random tree and change it around, you'd see that there's more entropy, and for a biological tree there'd be less entropy, because those cells are actually, they have to be in a certain place most of the time, or else they get development gets disrupted.
Okay.
I mean,
I gotta run now, so
okay.
Good seeing y'all.
Well, good seeing you.
I'll be checking in next week. Probably.
Okay, thank you.
Soon.
Bye.
Okay, bye-bye.
It looks like Morgan joined us, and Jesse and Harikrishna had to leave, so thank you for attending. So, Morgan, hi. Nice of you to make it. Are you... Oh, okay, there you go. Could you introduce yourself? I don't think anyone's met you in this group.
Sure. Yeah, hi, I'm Morgan. I am a neuroimager by training, and I've been joining the Saturday NeuroSim meetings for some time, and really interested in both computational models as well as developmental neuroscience. So been from last week's meeting, not catching the video. I followed up the worm base paper, just looking up kind of the resources and things that are available, which are impressive and broad. So yeah, it's been interesting to see, and I've been trying to lead a brain organoid, cerebral organoids interest group in the oral meetings, and that's kind of some of the overlap between the DevoWorm and things I'm interested in. So it's great to be here.
Yeah, nice of you to join us. We were just having a conversation about some things in developmental biology, and then we were talking about Google Summer Code for this group, which is, I know we have Google Summer Code in the other group, but in this group we have two projects. We have the graph neural networks, and then we have the a spherical map of the axolotl embryo. So those are, yeah.
Yeah, no, and that's great too. So, like I said, some of the overlap here is that I think any of the organoid work would require a good. Sure, it's my name at Gmail. So, but I can send it to you. So I like some of the microscopy overlap. And really interested in one, seeing what's available here in the Bay Area for independent people, but also wondering if some of the photogrammetry work that we've done previously might be useful for the photo stitching that I think you were doing.
Yeah.
But definitely would love to learn more about. I know that Chan Zuckerberg supports an image library for microscopy. So it's like something like N pairs or something. I know that's actually a different package.
Yeah.
but it looks like N pairs.
Yeah.
So I don't know. I need to learn more. The last thing I've heard about is Image J, and that was 20 years ago. So it's been funny to review all the kind of agent-based modeling software. It's like 70-80% of it is all Java, and I swear that's just because of the old Swarm packet, or
probably, yeah.
There was this Java ecosystem. Anyway, really interested in that, and of course the graph neural networks is in neuroimaging and kind of connectome analysis. Graph-based analysis is very hot these days, and I'm also really interested in overlap between areas that kind of research communities that haven't necessarily talked. So I'm really interested in the Neurodata Without Borders user days that's coming up, and really more. It would be interesting to look at some of the file format issues and things like that from invasive recordings versus EEG recordings. But it's really looking at some of the libraries that invasive people use that EEG people aren't really familiar with.
Yeah.
And really looking forward to that.
Yeah. So we've actually discussed photogrammy in the group here, especially with respect to this project, but also other projects. And in our YouTube channel, we have a couple of talks on different techniques of microscopy. We had one person in the group a long time ago who's since deceased, who was he did a lot of stuff with, he was an iron worker and all these other things, but he did some different forms of imaging like biological imaging and things like that. Steve McGrew, and he has a talk in one of our playlists on. I can't remember what the technique was, but we talk regularly about these different imaging techniques, and Dick, of course, he's done a lot of work on this in his career. So, and then some of the other stuff with organoids. I know that we've talked a little bit about organoids in the group, but it's been very, it's kind of like the problem you have with organoids is it's very difficult to get a sense of the landscape of how to use these for different things. So in this group, we thought about a little bit more broadly. I think we've discussed different organs and organoids and more kind of how they integrate with development than maybe with brain function. But it's a lot of really interesting problems.
I'd love to get an optical coherence tomography image of some organoids. I think that might be interesting.
Yeah, I mean the kind of microscopy, and these days it's multimodal microscopy, or I don't know what you want to call it. I think it's funny that the astronomers call it multi-messenger.
Oh, yeah.
Definitely more kinds of as well as some of the kind of clearance techniques and ways to kind of prepare tissue for microscopy or imaging.
Yeah, I like everything live. If I can.
Okay.
Yep.
I see. Yeah, sure. No, it's yeah. Would love to see more about that. I wanted to bring up the Morpheus package, Bradley. I don't know if you mentioned that before, but I mean, in trying to get more practical as well as try and think of things that perhaps others that are interested to get involved with. I've been thinking about smaller software projects and things like that, and this Morpheus package has a lot of nice simulations and models for both development, stem cells development, and kind of other biological processes, when Bradley and I have talked about goals for a cerebral organoid program, I should say Bradley's note was we need a computational model, can we make a computational model of this, and by that, and with OpenWorm looming in the background, I assumed they meant a very low level bottom up.
Yeah, well. You have to start somewhere, I guess, is the...
Yeah. No, exactly. Yeah. Start with the complete model.
Well,
see what you can reduce.
Yeah, there are a lot of models in a lot of computational models now for embryogenesis. I mean, they're not, there are varying degrees of usefulness for organoids, but the idea, that you can trace lineages, or the idea that you can look at different cell fates, and organoids aren't the same as biological development, but you could take some of those principles and say, I'm going to build a model that has these properties. The geometric aspect or the spherical aspect as well is very important. So, I guess they're not really, some of them are spherical, some of them grow in weird shapes, but that's something you want to capture, right? So, yeah, those are all important. So, I mean, And
why? And can you give us some backstory of why Max Tegmark is doing has a paper on 3D reconstruction of nematode neurons?
I don't know. Well, yeah, Max Tegmark is a physicist who's done a lot of stuff on different things, and he's I guess he put this paper out. I put it in the Slack channel but it's.
I mean.
Yeah, they basically reconstructed. What was that?
Has he been, doing stuff with nematodes for a long time?
I
didn't know that.
I don't think so, but nematodes are kind of like the best case scenario for a biological organism, like with terms of modeling things
Simple.
and self. Yeah.
Yeah.
So,
Yeah.
but this is something that I don't really know his motivation other than like maybe he just wanted to do it. But yeah,
Let's see. I just thought, I didn't know if that's like an older research program that I just wasn't aware of, or that's a new interest.
I'm not sure, but yeah, C. elegans has a really nice community of you were reading the worm-based paper. They have a really nice community of people who do stuff. So
And the Morpheus package has quite a few development zebrafish development.
Right. So that's something. Yeah, we've done a we've done a little bit of stuff with zebrafish data in the group. Not much because it's like, it's a different type of development, different type of organism, but it's not. If you're going to compare with other things, I think we've also done some work with sea squirts a long time ago. Dick and I did some work with, looking at the data from sea squirts. So there are a lot of data sets you can use, for comparative purposes. So some of the papers I was just showing you, they are sort of have this comparative aspect where you have two different species and they have very different development, but you can still gain insight from that because they have, they have processes of cell division, they have processes of cell differentiation. Those are things you can compare and say, there are these things that they have in common, these things that they don't have in common. So it's nice to have that comparative approach. So it's very much very useful, and I think you could do the same with organoids. If we had organoid data, we could compare it to some of the real data from real organisms. Or
Morgan and Bradley, I hate to argue with you, but I don't think nematodes are that simple. They have
Well,
about 900 different cell copies, okay, which includes 50 pairs of identical cells.
Yeah. They're not simple by any means, but it's definitely not like doing something with a mammal, I guess, is the.
I suppose an advantage is that the differentiation tree is about the same as the lineage tree.
Right.
It has many cell types.
Yeah.
Okay. Well, I'm going to kind of disappear into the background as I take my daughter to school.
Okay.
But I will be listening and
Okay. Well, I think we're done for. Yeah.
Okay.
So, thanks for attending.
Nice meeting you.
Nice meeting you.
Yeah, nice meeting you. Yeah. Bye.
Okay. Well, have a good week, everyone. All right. Bye. Hello. Now I want to go over a paper on body plan identity. This is something that just came out in the literature, and hopefully it's informative to some of our students, and it'll maybe foster some conversation amongst our other collaborators. So this is paper Body Plan Identity, a Mechanistic Model. This was published in Evolutionary Biology, I believe, this year. And it was March 21st. And it's by James DeFrisco and Gunter Wagner. Gunter Wagner has done a lot of stuff with theoretical work on robustness and evolvability. So this is an interesting perspective on body plan identity and development. So let's zoom in on the abstract here and let's go through this paper. So the abstract reads, a body plan is a stable configuration of characters for a major taxonomic group, such as chordates or arthropods. So a body plan is, of course, the sort of architecture of an organism. And in evolution, we see a number of body plans emerge quite early, actually in the Cambrian period. And those body plans have remained consistent throughout evolution. So you get a number of body plans that emerge in that several million years ago, 100 million years ago, and continue on through today with some modifications. So, for example, arthropods, which are these segmented invertebrate organisms like different insects, different crustaceans, organisms like that, they have this segmented body plan. You see this again and again across different species of that taxonomic group. Chordates being organisms with a spinal cord or with a notochord or with other types of central cords, nerve cords. They all have the same body plan where they have bilateral symmetry in this nerve cord. And so what they're saying is that this is a stable configuration of characters, meaning that it's a stable sort of configuration of traits and how they're arranged in space. So this is the approach that they're taking to development. It's very evo-devo. Despite widespread causal reliance on the concept for guiding comparisons within and between groups, the nature of body plans, as well as the biological causes underlying their evolution, have remained elusive. So we don't necessarily know why they've evolved in the way that they did, and we don't know why they remain stable. We just know that they do. So it's not really easy to figure that out. We just kind of make this assumption that, we can see in the data that it's remained stable. We don't exactly know why, other than to say it's been conserved. A lot of people will say it's conserved in the sense that it doesn't change. But why doesn't it change? This paper proposes an abstract mechanistic model of body plan identity. So they go into this idea of positional identity, or a certain type of developmental identity, which requires the cells to have an identity that is comparable between cells and that puts them in a certain order. So we've talked about in different group meetings, we've talked about positional identity and how cells have an idea of kind of where they belong in the embryo, where they should migrate to, and what they should do if they're in a certain position in the embryo. Should they become one fate or another? And so this is building upon this basic idea. We hypothesize that body plans are an evolutionary phenomena that only applies to a relatively small subset of major clades, and clades are evolutionary groups in a tree, rather than being associated with each and every so-called phylum. And so this is, where you have different body plans in different parts of the tree. They don't necessarily map to our nomenclature that we use in science. They map to organisms that descend from a common ancestor, and whether those be in any defined phylum or not, which is just the name that we give a group, taxonomic group, isn't really the point. Body plans arise in evolution by stepwise accretion and require a level of developmental complexity that is only found in some animal clades. Further, we suggest that, parallel to the developmental mechanisms controlling character identity, that there are body plan identity mechanisms, or BPIMs, that maintain entire configurations of characters while possessing a mechanistic architecture that is itself stable and traceable through evolutionary change. So this is, again, this idea of these mechanisms that enforce body plan identity. They maintain these different configurations, so they maintain different segments of the organism. They maintain this bilateral symmetry, and they do this in a way that's consistent and that you can trace through evolution, and that's stable through evolution. These BPIMs, we suggest, are entrenched intercellular signaling networks operating between a transient embryonic structure or different embryonic structures that are destined to differentiate into distinct individualized characters. So again, this is they hypothesize that they're part of these intercellular signaling networks. So a signaling network is something that allows cells to communicate. Intercellular networks allow cells to communicate with one another. They secrete different signaling hormones. They secrete different types of signals, chemical signals, electrical signals, other things that allow them to communicate with their neighboring cells. And when cells exist in a group of cells together in a certain location, they can take on a certain fate. So if you take a stem cell and you put it in the middle of a bunch of liver cells, that cell is going to become a liver cell where it'll die. And so that enforcing differentiation mechanism, is actually quite important to maintaining the integrity of an organ. If a stem cell is dropped in the middle of a bunch of liver cells and doesn't become a liver cell or doesn't die, it can become a cancer cell, and it can invade the entire organ or group of cells that it belongs to. So these are things that are actually quite important in development and maintaining healthy tissue and maintaining biological homeostasis. The activity of a BPIM results in a transient long-range integration of the embryo that is highly sensitive to genetic and environmental perturbations. And that can be detected morphologically as a conserved phylotypic stage. So the phylotypic stage is a part of development where everything is really conserved, and that you get the phylotypic, meaning it's that phylotype, it's this specific type that the embryo conforms to. It's a period of development where there's a lot of conformity, general conformity, and then later on you get this. People have talked about the hourglass of the phylotypic stage. The phylotypic stage being at the center of the hourglass, the thin part of the hourglass, the early part of development being beneath that, and the later part of development being above that. And so this phylotypic stage sort of enforces a lot of these mechanisms. And it also allows the embryo to be sort of robust to some of these environmental and genetic perturbations. But the BPIMs give you this long-range integration spatially in the embryo that can actually be sensitive to genetic and environmental perturbations. This model is illustrated with detailed interpretations of Notochord signaling system and the segment polarity network as candidate BPIMs. So two examples of BPIMs is the Notochord signaling system and the segment polarity network. Segment polarity network we know from the world of Hough genes, which are these collinear genes that are expressed in a certain sequence, and that their expression factors actually play a role in building the segment polarity network. But the polarity network also relies on this intracellular long-range signaling between cells that tell them basically where to go, how to set up their sort of pattern formation, and so forth. Similarly, the notochord signaling system is similar, how to set up where the notochord is. And so there's a lot of this sort of signaling in these two examples. So these BPIMs you can see these in vertebrates and arthropods respectively. You see notochord signaling in vertebrates, segment polarity in arthropods. We conclude by contrasting the proposed developmental mechanistic conception of body plans with the phylogenetic notion of ground plans. So they're introducing a new piece of jargon here. Instead of body plans, they're using the term ground plans. And so this just kind of brings it more towards a phylogenetic interpretation rather than a developmental mechanistic interpretation, and sketch the general outlines of an empirical research program in body plan evolution. So this is so they kind of review the problem here in the introduction. They talk about some of these, so the origins of the body plan as a theoretical construct and as an empirical construct. So a curious evolutionary feature of body plans is their entrenchment. Once they involve, further evolutionary change seems to be largely restricted to modifications of that basic design rather than transformation into a novel or existing body plan, except in cases of major reduction in body complexity, such as in parasitism or some other situation like that. So this striking phenomenon is classically framed as a problem of understanding why the major animal body plans have remained largely static since the Cambrian. So this is, again, hundreds of millions of years ago we get these body plans, and then we stop evolving body plans. So why don't we just evolve infinite body plans? And the answer, of course, is not necessarily well known. We're trying to figure out exactly why that is. Some people think that there have been novel body plans that have appeared since the Cambrian. Holometabolin larvae being an example of that. So this is in insects. There's maybe an example of this. In any case, there is much more to the concept than a puzzling evolutionary pattern to be explained. So this is why they're doing this reinterpretation. They talk about sort of the details of the body, the concept of body plans, some of this mechanism. So with this end, all right, so they talk about the positive role for the concept is significantly enhanced by mechanistic models of the interdependencies in development that give rise to evolutionary conservation of body plans. Such models would contribute not only to explaining macroevolutionary patterns of body plan conservation, that is, between species, between groups of the phylogenetic tree, but also to understanding the possibilities and constraints on the evolvability of specific body plans. So understanding how those evolve and their capacity for evolution, while also refining and clarifying the body plan concept. With this end in view, we propose a conceptual model of body plan identity mechanisms, which are those BPIMs, building on the framework of character identity mechanisms. So this is something that they've I've worked on previously in more of a phylogenetic context, using characters or characters being traits that evolve. You can identify a basal version of it and a derived version of it. The derived version is usually what it's evolving towards in different lineages. So you have many derived forms and a single basal form usually. So this is the way they're kind of approaching this. Together with work on the developmental hourglass model from Rudy Raff, who is a famous developmental biologist. And this hourglass model is the one I mentioned, where you have this phylotypic stage in the middle. You have early development, late development. And early and late development is where you have this vulnerability to different environmental and genetic perturbations. The phylotypic section, part of the hourglass, is where things are largely conserved. And this is somehow related to this generation of body plans in development. So they actually propose two models. They have a developmental mechanistic body plan concept and the phylogenetic concept. And so if you take that extra phylogenetic view, you see that you can actually get a little bit more out of this concept than you otherwise would. So again, body plan identity versus state. They talk about this. They talk about the concept of homology, which are shared derived characteristics or characters. And following the parallel with homology, we propose that there is a biologically meaningful distinction between body plan identity and body plan state. So these are two things, identity and state, which means that there's an identity, which is where there's it's like you belong to some group, versus a state. Are you in that group currently? And so there's this discussion by Günter Wagner on character identity and character state in evolution. So there are these phenotypic characters that they're trying to sort of understand the state versus the identity. So you're identifying a character versus having a state of a character. So we can identify something as having a body plan, right? Whether it has a certain body plan or not, and then whether that organism actually is in the state of that body plan. So, there's a lot of variation in body plans, although you can identify them by looking at different organisms that have them, but they're not always in the same state. And so you have to distinguish between both of them. So the character identity of the tetrapod forelimb, for example, refers to its identity as a body part, which can exist in many different character states, in particular tetrapod lineages. So you can have wings, legs, flippers. They're all basically forelimbs, and those are states of forelimbs. But the actual identity of a forelimb persists across all of them. So you can have different forms emerge, and you can still identify it. Character identity is based on the relative position of the character within the body plan, as well as the other operational criteria traditionally used to establish homology. So they get into this long discussion of different, mapping this to sort of a phylogenetic approach. You're talking about positional identity in some cases, but you're also talking about positional identity in the context of these different different parts of the body plan. So, for example, a trait or a cell must be in a certain place. So the forelimb must be towards the front or the anterior end of the organism. A forelimb doesn't just form out of the tail, or it doesn't form in the back of the organism. So the cells have to have positional identity to know that they're going to be part of the forelimb. But then that forelimb itself can take on different forms. It can be a wing, it can be a flipper, and that's part of the sort of specialization locally of the cells. What do they become? What does their intercellular signaling tell them to become? So they kind of go through some of the history of this. The body plan is not the same as an idealistic archetype concept, which is something that's been around for since pre-Darwinian times. The body plan is a much more refined concept, but it's still not, even though it's post-Darwinian, it's not really been incorporated with phylogenetics. So they're kind of they're identifying mechanisms. They identify these character identity networks, which is something that Gunter Wagner came up with in the past. This involves gene regulatory control of character identity. So now we get into gene regulatory networks, and of course this links to the Hough gene, the collinearity of Hough genes, and other genes like UBX, which controls hindwing character identity in insects. Those genes then exist in networks, and you can knock down certain genes and show that they have developmental defects, but you can also show that they modify that body plan a little bit. Actually, they don't necessarily modify the body plan. They change some of the traits in that body plan. So they do. There is this aspect of sort of modifying the character state, but you do have this. You still have the basic body plan, even if you do something like that. So it's a very interesting relationship. These sorts of studies with the different genes, knocking out genes and showing that they're, additions or deletions in the phenotype, those are classic evo-devo experiments. And linking those to this sort of phylogenetic approach, I think, is quite valuable. And they kind of go through these character identity mechanisms for anatomical units at different levels of organization. So for cell types, you have gene networks, you have transcription factor complexes, non-coding RNA. For tissue types, you have cell types and extracellular matrix. And for organs, you have signaling molecules and signaling centers. So these are different ways that you can identify these character identity mechanisms at different levels of organization. And, you can make that connection between that and the phenotype and the body plan. So I'm looking to see if there are any nice figures in here, and I was kind of hoping for a diagram or two. Here's a diagram of key components of the notochord signaling system. So this shows the notochord as it's forming in the different tissues, and how they're all sort of in this sort of arrangement. And this doesn't vary too much across species. So things have to be in a certain anatomical location. And then you have, aside from this anatomical organization that's conserved, you have these different signaling gradients. So you can see that there's signaling gradients that set up these different parts of the body. So the BMP gradient and the WNT gradient start at the anterior end and fade towards the posterior end. The SHH gradient is both sort of anterior, posterior, and towards the middle you get a higher concentration. So as you reach the midline, you get a higher concentration of SHH. And all these things, these gradients set themselves up to enforce this body plan in a developing organism that has this body plan available to it. So let's see if there are any other figures here. There's another figure where we show the segmentation body plan. So this is in arthropods. This is where you have this simplified illustration of the SPN showing the basic positive feedback circuit. So this is SPN. I'm not sure. I don't think we encountered that abbreviation before, but this shows basically the formation of these parasegmental boundaries. So you see in the arthropod, you have these different segments And in between the segments, you have these parasegments that show up in the body plan. And these are repeated across the body plan, anterior to posteriorly. And you have these small regulatory networks that have this motif. So they always have the same sort of motif. They regulate each other in a certain way. And they regulate themselves across the segments. So each segment has the same effect. They have an internal regulatory network, and they have this intercellular regulatory effect. So you have HH and WG, and they're regulating their neighboring segments, and they're basically setting up this gradient across the segments and communicating with each other to make sure they're in the right position. So there's a lot of, so they go through this model as well. And there are a lot of nice references in this paper. As I'm looking through it, I'm noticing this. And then so finally, the discussion is body plans beyond typology and genealogy. So they kind of get into these mechanisms. They use these two examples. Their central hypothesis is that these BPIMs have the same basic architecture and causal profile as character identity mechanisms for organs. So these are cell-cell signaling networks characterized by active modularity, which means components that are distinct, complex organization, collective necessity, and non-redundancy. In the BPIMs, however, the tissue domains covered by these signaling mechanisms are weakly individualized, transient embryonic structures, often distinct from germ layers that are destined to differentiate into distinct individualized characters in organs under control of different CHIMs. So this is a linkage between these BPIMs and CHIMs. So this is so that's all I wanted to talk about with this paper. Hopefully this was educational. If you have any other questions on that, please let me know. Thanks.